Hello. Each of our cells have about 23 chromosomes and 23 pairs of chromosomes, and each of them is a double-stranded DNA molecule, and they're very long. In that sense, one could expect that there is occasional breakage, and then the number of molecules, if you see, the double-strand breaks could happen, and then the number of DNA molecules could be increased. And what happens in that situation? There are at least two ways that we know of how those kinds of double-strand breaks are repaired. One is using recombination, because there are pairs, there are homologous recombinations can happen between the homologous chromosomes, and repair could happen. But there is also another one that is called as non-homologous end joining, and that is what we will discuss now. I am Dr. BCMR. In the non-homologous end joining, the double-strand break is recognized as like this, and that is taken up as, and there are two proteins called as Ku70 and Ku80. They recognize and encircle the ends, and it's important to remember that if there are double-strand breaks, there is going to be cleavage, or exonuclease might act on it, and then the whole chromosome is going to be lost. In that way, the cell is going to have detrimental effects on the cell. So these ends should be sealed and rejoined so that the cell can exist as long as it should. So the double strands end, the ends of the double strand breaks are recognized by Ku70 and Ku80. Of course, in a chromosome, at the ends we have telomeres, so they are not recognized as double strand break points. Instead, if a break happens in between, that is where telomeres are not there, and there are several proteins which bind to telomeres and they are not there, so it must be those ends must be the products of a double strand break. So Ku70 and Ku80, they bind to such ends, and that then they will allow the recruitment of protein kinases, DNA PKCS, and along with an important protein called as Artemis. Artemis is a protein that processes the ends, and it has exonuclease and endonuclease activity, and the ends that are present are going to be processed so that they will be ready to be ligated. Of course, because of this processing, some genetic information might be lost from these ends, and that is why this non-homologous end joining, or NHEJ, is error prone, because we are losing some of the information from these ends during the processing. And ultimately there will be recruitment of other proteins like the ligase, XRCC4 and others, and then they will allow the fusion and sealing of these two. This double strand break is repaired. Yes, there is a loss of information, but at least the rest of the chromosome is fine. If this kind of double strand break happens within the intergenic region or neutral sites where there is no essential gene produced or silenced DNA, then the effects are not going to be as much in that particular cell type at that point of time. That's how non-homologous end joining is still, even though it is an error-prone one, it is still an useful and very important repair system that is present in eukaryotes. In prokaryotes too, there could be, but majority of the time the homologous recombination is the way of repairing double strand breaks. I hope that is clear. Thank you very much. See you later.